So this is the official title of my presentation, but in my department, we like to give it a slightly different name, the Quest for Functional Variants in Human Endogenous Retrovirus. And hopefully by the end of this presentation, all of you know what is a human endogenous retrovirus. I think that we as people, as we go through our daily lives, we try to maximize the amount of information that we have access to. For example, even when we're walking, I think most of us have two working eyes, but we don't go around with one eye closed. Why? Because you might miss important information such as a rock, a chair, a step, or something. And I think this is especially important when you're trying to solve a problem, because having that extra piece of information might actually make you look at the problem in a different light, and hopefully that will help you solve the problem. But if we apply this standard to something as simple as walking, why do we accept only using 50% of the DNA when we're actually making decisions that will impact the patient's life? Especially when the other 50% of the DNA still has many functional and regulatory elements that will impact the disease prognosis and progression. And in essence, this is the goal of my project. We want to expand the percentage of the DNA that we actually use to make clinical decisions, and hopefully this will help people. In particular, we're interested in looking at this set of sequences called human endogenous retroviruses, or HERVs for short. These correspond to nearly 8% of the genome. They are categorized as repeats, and more specifically as interspersed repeats. And quite interestingly, these are actually ancient viruses that infected the human genome millions of years ago. But you might ask, why are we interested in these? Well, they have been associated with many diseases, including multiple sclerosis, amyloid lateral sclerosis, and different biological processes, such as embryogenesis and aging. But in our department, we're specifically interested in them because of their association with cancer. Many studies have associated HERVs with cancer, even so far as there being databases just to registering the association between these. Furthermore, a paper that was published actually this year showed that patients that were being treated for melanoma using checkpoint inhibitors actually had a differential survival based on their level of HERV expression. For example, on the plot on the left, you see on the blue line, patients that had a high expression of HERVs actually had a lower survival than patients that had a lower expression of HERVs with the red line. And this difference was especially more pronounced in patients that had a high tumor mutational burden, as can be seen on the right plot, which means that they just have a lot of mutations in their genome. But the exact relationship between cancer and HERVs is still not quite known. We don't know if they actually have any impact at all, if they are maybe one of the causes of cancer, or if they exacerbate it. But early results can already point us to the fact that there may be a causal relationship. Here, a plot plotted by my supervisor, Falek Utzenbarger, we can see on the y-axis the total expression of HERVs across patients, and on the x-axis, we see different cancers. And what we can gather from this plot is that the distribution of HERV expression across cancers can be quite different. And this points us to the idea that maybe they do play a functional role. But to be able to progress with the research area of HERVs, we need to be able to actually identify what they are. And over 50 years of research, researchers have come up with this theoretical model of what a HERV is with all these different components, the LTR, the long terminal repeats, and different viral proteins that they should have, GAG, PRO, POL, ENV. And although this is a very beautiful model, in practice, you rarely see these. When you look at actual DNA sequences, what will happen is that most HERVs are going to be completely disfigured. Many of those will have missing components, and even when they have these components, the components are completely filled with mutations, including frameshift mutations and stop codon mutations, aside from also having different variants, including SNPs, indels, and structural variants. To further this problem, there's the problem that there is no one standard way of defining or identifying what a HERV is. Different people in different studies will use different methods, and this actually makes it quite complicated to translate the results from one study to the next. For example, we have methods that simply use the annotations for the reference genome. We have libraries with consensus sequences, libraries for profile hit and Markov models, and even de novo structural detection methods. Another thing that we need to consider is that HERVs are a very broad group of sequences. Again, these correspond to nearly 8% of the genome, and so we cannot just consider them as a single group. We actually have to categorize them into different parts. And there was throughout history, a lot of different ways to categorize them, and the most recent, in 2016, they used sequences that were obtained by retrotector, and then categorized them based on phylogenetic analysis. They were categorized in 3 classes, 11 supergroups, encompassing 40 canonical groups and many, many subgroups. And in our department, we think that this may be the key for continuing the research. We believe that although this categorization method is quite logical, it makes a lot of sense, we believe that we can do better. We can go a bit further. And instead of categorizing them based on their phylogenetic relationship, we believe that we can categorize them based on their functional relationship. And this would hopefully help us identify better cases in clinics where maybe the difference between one patient and the other one is maybe the position of a HERV in a specific context, or having a variant or not within HERVs. But before we can suggest or provide a new method, we actually need to look at how people have been defining them. And originally, HERVs were identified by biologists grabbing the DNA of viruses and trying to match in the human genome sequences. And whatever they matched, they would call human endogenous retroviruses. Then the transition changed to primers, to PCR primers. And PCR primers are simple, small nucleotide sequences, usually between 10 and 20 nucleotide bases, that are used to target specific portions of the genome and amplify it so we can measure and sequence them. And this definition is still used to this day. So in order to study, we decided to compile a database of over 500 primers that have been and are being actively used in the literature. This database includes many of the HERV groups that were categorized based on phylogenetics, and we can see an overrepresentation of HERV-K. Why? Because this is the most recent out of HERV-K and is thought to have the most functional role. Because the HERV sequences are so broad, they're around 10,000 nucleotides, you cannot design primers to amplify the entire region. You usually need to design primers to amplify specific portions of the HERVs and specific components. And in this database, we were able to collect enough to amplify different components of the HERVs. However, the primers cannot be used by themselves. We need to align them to a genome so we know what they are actually amplifying. And we decided to, instead of relying on the linear reference, we decided to build what is called a graph pangenome. Why did we use this? Because it's been shown to provide better and more accurate results associated with indels, SNPs, structural variants, inversions, and other types of variants. But what is the graph pangenome? Well, a pangenome is nothing less than more than just a collection of genomes. And a graph pangenome is a data structure that represents a pangenome in a more compressed format where the common parts are compressed, but it still allows to show all the diversity between the different genomes. We built this pipeline using publicly accessible data from Genome in a Bottle and Platinum Pedigree, which they had PacBio HiFi reads available, and we used the tool HiFiasm to reconstruct what their genome should look like based on their reads. We also included some assemblies already made by the Human Genome Structural Consortium, and using simply the Cactus Minigraph Pangenome tool, we were able to build our variational graph pangenome. By aligning the primers to the pangenome, we can see that actually the primers map to many different locations, meaning that they are high ambiguous. What you can see in this plot is just the number of alignments that each primer has, and then they're sorted. What we also see is that the ambiguity of the HERV primers, it's not specific to any of these groups, meaning that they always match to multiple positions independently of which category of HERVs they are from. But we also see that the type of ambiguity is dependent on the HERV group. So some are more ambiguous than others. And we can see the same thing for primers, for components, that it is common for all components, but it's not specific to any components. Again, we are looking at different methods of how people have defined HERVs, and one of the most used is based on the HERVD database. And one thing we can already see that is quite similar to the primer database is that even the HERVD consensus sequences are quite ambiguous. And this can be a bit of a shock since the HERVD consensus sequences are actually quite big. Whereas the primers were only 10 to 20 base pairs, the HERVD consensus sequences can go from 500 to even 1,000 base pairs, and they are still ambiguous. And again, this ambiguity is not specific to any of the HERV groups or any components, meaning that they map to multiple positions in the genome. We are also interested to see how are these sequences expressed. And when we look at this plot, I think it might be a bit of a negative result in the sense that most of these HERVs are not expressed in healthy cells. But one thing you need to remember is that these are 8% of the genome. And so even if there's only a few that are expressed, the few are still 100,000 sequences. Again, we see that the HERV expression is not specific to any group and is not specific to any component, meaning that we cannot just predict their expression based on the categorization. So we cannot predict their regulatory function based on the phylogenetic characterization. We can also look back on how does the ambiguity of the HERV sequences affect the mapping. And right now, with the simple plot, we can have an idea that the expression of the HERVs, even if they map to ambiguous sequences, they do have similar-ish expression profiles. A logical question to ask is, does this database agree, the primer database agree with the other annotations? Specifically because we're not interested in just using primer annotations, we want to develop a better annotation method for HERVs. And what we can see is that the primers do align to locations where the HERVD consensus sequences align to, but the primers, on the other hand, only overlap with a very few of the HERVD sequences, meaning that in most studies, we're actually not studying the vast majority of HERVs that exist. But these are only two methods of annotation, and we actually want to expand this a lot more. I've mentioned earlier that there are methods based on profile hidden Markov models, such as DFAM. We know that there are other methods that were used to originally categorize the data based on retrotector that we also want to encompass within our annotation method. However, these methods were not made for graph pangenomes. They were made originally to deal with linear references. So we want to adapt these methods so we can provide better annotations for the graph pangenome. But again, this is only part of the overall project. We don't want to just give a standard way. We don't just want to recategorize. We want to be able to fully characterize the HERV sequences in respect to variants. We want to be able to say which roles does each HERV play, and then check which variants within each HERV affects the disease outcome and disease prognosis of individuals. With this, I would like to thank HiTSeq and Hervolution, with a special thanks to my supervisor, Falek Otzenbagger. Thank you.
Thank you. We have time for questions. Maybe I will start. So you were studying the primers, and so is the goal to also design new primers that will be more sensitive to these unseen HERVs, or what was the primary driver for looking at the primers?
The reason why we started looking at the primers was it was the most common definition of HERVs, and we were in doubt if people were actually observing what they said they were observing. I think based on this research, it is possible to design better primers, but that is not the only goal. We want to be able to fully characterize the diversity that exists in HERVs and how they relate to different diseases.
And so going forward, you probably more envision studying like genome sequences or like full genome sequences, or do you still think that the PCR might be useful? Or
I think overall the best method is using long reads, but it is still up to question how much. I think based on the graphene genome, we can have a very good idea of what kind of percentage of how good the primers can. Yeah, it's a bit tricky. Sorry.
okay.
Yeah.
Okay. So are there any other questions? It seems not. So let's thank the speaker and all the speakers of this session.